Hello, we're glad you've joined us for this live webinar, Allergen Detection and Authenticity Testing of Food using Targeted Proteomics and MRM Cubed. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by SciEx. SciEx helps to improve the world we live in by enabling scientists and laboratory analysts to find answers to the complex analytical challenges they face. To learn more about SciEx, please visit sciex.com. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose past questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speaker, Jens Brockmeyer, PhD from the University of Stuttgart, Department of Analytical Food Chemistry in Stuttgart, Germany. Dr. Brockmeyer studied food chemistry at the University of Münster in Germany and received a PhD in the field of protein biochemistry, molecular microbiology, and molecular biology of enterohemorrhagic E. coli in 2009. He was an independent research group leader in food proteomics and allergenomics at the Institute of Food Chemistry in Münster and a visiting postdoctoral research fellow at the Manchester Institute of Biotechnology in Manchester, UK. Since 2016, he has been a full professor of analytical food chemistry at the University of Stuttgart. The major focus of his group is the development of novel analytical approaches for the characterization and detection of food allergens and the authenticity of food. Dr. Brockmeyer will now begin his presentation.
Thank you very much for the kind introduction, and I'm happy to present you some of our research and some of our data on allergen detection and authenticity testing of food using targeted proteomics, so mass spectrometry, and especially also a approach which is available via SciEx Q-Trap instruments, the so-called MRM Cube technique. But before I'd like to go ahead and show you the experiment and the mass spec data, I'd like to give you a brief introduction on authenticity and the interconnection actually of authenticity and food safety. You all might be aware that in Europe the horse meat scandal really was of huge importance concerning food authenticity, and since then there is a coordinated focus on the detection of food fraud by Europol, Interpol, and these are the so-called OPSON operations, which take place every year, and this year it was the OPSON 6 operation. And the focus in Germany was on hazelnut for specific reasons, because the hazelnut harvest was not great in 2016, which means that the hazelnut prices go up, and the available amount of hazelnut, of course, were lower this year, and this increases the likelihood that food fraud occurs in this special food. And the authorities really detected huge numbers of fraudulently blended hazelnut products. And the problem in this case was that they observed blending with other allergenic foods such as peanuts, cashew, almonds. And the problem is that peanut allergic, for instance, might tolerate hazelnuts, and if you do have non-authentic or fraudulently blended hazelnut products, this can really then be linked to food safety, as you might see allergic reactions. So at the first glimpse, both topics, authenticity and food safety, seem to be separate, but there are regularly incidences where food authenticity also affects food safety. What I would like to present you today is the use of mass spectrometry as a tool for species authentication or for authentication in general. And one example is the authentication of different species, so the distinguishing between mammalian species, for instance, marine foods, also plant species. And what you would like to have is you need biomarker peptide specific markers which you can use in your mass spectrometer as a marker for your given species. So the first thing you have to do when you want to use proteomics for species authentication is to identify species-specific sequence polymorphisms. And when it comes to allergen detection, then you do need allergen-specific sequence polymorphism to specifically detect marker peptides that allow you to identify these food allergens. We do not analyze intact proteins because this is really in complex matrices such as food pretty demanding and in some cases not feasible. Therefore we digest the proteins or the complete proteomes, the complete set of proteins using specific proteases such as trypsin to generate peptides and identify from this huge set of peptides then the marker peptides. The next step is then the validation of specificity. You can do so by using databases, but the problem is that databases are far from being complete. Therefore, we in general perform experimental validation of specificity. So we get hand at authentic samples and really do see whether the identified peptides are really valid and specific on experimental basis. And there are, in addition, some analytical problems where sensitivity also might play a role, and therefore I'd like to give you also some examples on optimization of sensitivity. On this slide, you can see the sequence homogeneity of mammalian proteins. It's exemplified by myosin 4. It's an abundant muscle protein and the partial sequence alignment between 5 different species: pork, beef, human, horse, and lamb, as you can see here on the left side. And it's only a partial stretch of the complete protein. The complete protein has something around 1,700 amino acids. And all the rest, which I've not displayed here, is completely identical. So the sequence identity of proteins is often around 95 to 98% in mammals, and this means that the identification of species-specific sequence polymorphism is a pretty rare event, and in this given example, in the complete stretch of this 1,700 amino acids long protein, you will only find one tryptic peptide which is suitable, here given in this red frame. So species-specific sequence polymorphisms are pretty rare events, but on the other hand, we are lucky that the proteome, the complete set of proteins really is full of different proteins, 10,000 different proteins in every mammalian cell, and around about 20,000 different proteins in the complete organism. So we have many, many chances to identify species-specific sequence polymorphisms. And in addition, mass spectrometry is nowadays able to detect and identify peptides in highly parallel fashion. So we are able to characterize proteins and proteomes in a very highly paralleled fashion. And this is in general done by the so-called bottom-up proteomics approach, which some of you might already have heard of. I'd like to give a brief overview for all of those of you who might not be familiar with this technique. So the idea is basically to build up the protein sequence and the proteome, if you want so, by digesting the proteins using specific proteases such as trypsin to the peptides. identify the peptides and build up the sequence from bottom up. Therefore, this name bottom-up proteomics. So what we are doing is we extract our proteins, perform a proteolytic digest, specifically in general using trypsin, leading to a vast number of peptides that are specifically released by the protein. The next step is then to separate peptides using standard chromatography, in general C18 material, which really nicely works with peptides in contrast to proteins, where chromatographic separation is often really a problem. And the next step is then MS and MS-MS characterization of these peptides. And one of the huge advantages of peptide mass spectrometry is that the resulting fragment spectra are really nicely reproducible and they are predictable. So if you have a look at the part here on the lower left, just indicated by this arrow, you can see that in general peptides tend to fragment at the emit bond, meaning that you generate so-called B or Y ions if the charge resides on the N-terminal side. After fragmentation of the peptide, you have a so-called B ion. If your charge resides on the C-terminal side, you will end up with a so-called Y ion. So under ideal cases, you will have a complete set of B and Y ions which describe your sequence of your given peptide with two-fold sequence coverage. And as the proteolytic digest and the MS-MS fragmentation is reproducible and predictable, you can use protein databases to perform a theoretical digest, ending up in silico peptides, theoretical peptides. You can then perform simulated MS-MS experiments, ending up in theoretical MS-MS spectra. And you can then compare the experimental data with the theoretical data, and by statistical means such as spectral matching and confidence scoring, you can with statistical confidence then identify your peptides, and from these data then also identify your marker peptides. And this is basically what we have done. One of the really major and most important points when it comes to experimental identification of peptide markers is that you have authentic material. You cannot only just go to the supermarket, buy any samples. We tried this several times and also saw fraudulent material, so you really need to be careful to get authentic material. You then perform protein extraction, tryptic digest to get the peptides at hand, separate your peptides using in general standard HPLC separation techniques such as on reversed phase C18 columns. Use high-resolution mass spectrometry to identify as many peptides as possible by use of database searches. And as the databases are in many cases for different foods are far from being complete, also de novo sequencing to identify as many peptides as possible. And one of the huge advantages nowadays concerning proteomics is that we are able to do this in a highly paralleled fashion, as I'd like to show you on the next slide. First of all, I'd like to give you a brief example on how these mass spectra can look like. So this is one example of a given peptide here on the left side, and you do see that we have nearly complete set of B and Y ions of this peptide, and here's the mass spectrum. The B and Y ions are annotated, so we have nearly complete two-fold sequence coverage of this peptide, and with high statistical confidence we can identify these peptides. And this is done in a highly paralleled fashion, as it is indicated on this slide. This is a 90-minute gradient of a partial the scrofa, which is pork meat extract, so it's not the complete proteome, but only a partial extract of some of the proteins from pork. It's the really abundant proteins, the myofibrillar proteins and the sarcoplasmic proteins, which really form the muscle fibers, are extracted in this experiment to identify marker peptides. And you do see that every dot which is represented here in this graph represents an identified peptide in this 90 minutes run with statistical confidence over the threshold which have we chosen for peptide identification. So these are the peptides which have identified with high confidence actually. And in this case, we identified from this partial extract something around 6,000 peptides. We had a pretty recent proteomics experiment just this week where we used marine foods and different fish species, and using complete extracts, we were able to identify in a single run 47,000 peptides. So this is really a highly paralleled approach to identify the peptides, basically. So we identified in this first approach using the partial extract from the pork muscle something around 6,000 peptides from around about 500 different proteins. Bear in mind there are something around like 10,000 proteins in one single mammalian cell, so this is really only a partial extract which we have used. The next step was then to search all these peptides against a database which we have generated from the available genomics and proteomics data from all species which are important for food purposes, at least in Europe. So we have included, of course, horse, chicken, lamb, different bird and fish species, and so on and so forth, and search all the peptides against this database to exclude all peptides which are not specific for pork in this case. And at least according to the database, there were 500 peptide candidates that were, according to database situation, might be specific for pork. And we have then some critical parameters which we apply on all of these peptide marker candidates. It's, for instance, the amino acid composition. So we try to avoid methionine and cysteine, for instance, because these are amino acids which are readily oxidized during food processing, leading to mass shifts, for instance, by methionine oxidation, and then you will not see this peptide any longer if you have a targeted approach. Similarly, we avoid post-translational modifications or missed cleavages, as these aspects also influence reproducibility. This, of course, further reduces the number of suitable peptides. We then end up with something around 75 peptides, which are in general suitable and fulfill all criteria. And the next really huge advantage concerning mass spectrometry of peptides is the predictability of the MS-MS fragmentation, because what we can then do is switch from high-resolution instrumentation to triple quadruple instruments and develop a method for all 75 peptide candidates on this MRM method and on a Q-trap or a triple quadruple instrument without having analytical standards for all the peptides. So we can do this method development right from the authentic sample. And there's a specific dedicated software around which really does an incredible job, and it's on top it's freeware. It's Skyline, developed by the University of Washington, which is freely available and is really an incredible piece of software. You can just use your peptides of interest. Skyline calculates the MRM transition because the fragmentation behavior of peptides, once you do know the peptide sequence, is nicely predictable. So Skyline then annotates the precursor ion mass and the expected B and Y ions, the MRM fragments which are to be expected, and then optimizes in an iterative fashion the single MRM transitions and the detection of the single peptide candidates by measuring all the peptides throughout the chromatographic run directly from your authentic food material, from your pork muscle extract in this case. And the major parameter which is optimized is in general the collision energy, but there are also other parameters such as declustering potential, which is also optimized. And by iterative measuring of your sample, you can then optimize all the peptides. Because you have switched from high resolution MS to lower resolution MS, of course you will lose further peptides that are specific in high resolution conditions, might not always be specific when you use lower resolution mass spectrometry. And in addition, we did the experimental cross-check, so we did not only optimize the method, but we checked all peptide candidates against other relevant species. So we measured also lamb samples and beef sample, horse samples, and so on and so forth. So this, of course, further reduces the number of peptides. We end up with something like 10 to 20 peptides, which are obviously really specific and can be seen using MRM mass spectrometry approaches. And we then simply choose the 5 to 10 most sensitive and specific peptides for the final method, and then use these peptides for our final method to detect the species of interest. And this looks like given on this slide. So there is one example of 3 different marker peptides. The marker on the left side, marker 1, is specific for pork and horse, and you do not see any signals for the beef sample here at the lower lane. In the middle, marker 2 is specific for pork, and you do not see any signals for horse and beef. And on the right side, you have a horse-specific marker, and thereby you can differentiate the different species and detect really the different species by using these markers. If we switch to other species such as chicken or lamb, you do see again nice specificity. What you sometimes do see, for instance, here for this lamb sample, that you have single MRM transitions popping up at shifted retention time, so unspecific single MRM transitions, and this is simply due to the complexity of the sample. If you have to keep in mind that from a complete extract from one single species, you will have something around like 10,000 proteins at least. And if you digest these 10,000 proteins, you will end up with at least several hundred thousands of peptides which elute from your column in something around 20 to 25 minutes chromatographic run. And from time to time you have, of course, a precursor ion that has very similar precursor ion mass than your peptide marker. And it might also be that 1 or even 2 of the MRM transitions also fit. Therefore, the minimum quality criteria which we use to really identify and detect a marker is that we want to see retention time fit and at least 3 MRM transitions, and the signal intensity ratio of the 3 MRM transitions also needs to be fitting. This is the minimum amount of parameters which we want to see to be sure that we really have detected this peptide. And in general, we use several peptides for the given species, so that we have not only one peptide marker, but a number of different peptide markers which we detect in our run. The next step was then to increase sensitivity, and and specificity as well, as I'd like to show you later on also for the allergen detection. And therefore we have used a relatively novel technique, the so-called MRM cube approach, and I assume that most of you are aware of what MRM actually is. You have your triple quadruple instrument, and in the Q1 you have the isolation of your precursor mass. In Q2 the collision cell, you fragment your precursor to the given fragments. You have your MRM transitions. And in the third quadrupole, you detect your fragment ions, your B and Y ions. And if the third quadrupole is a linear ion trap, you are able to, first of all, accumulate your fragment ions and then perform a secondary fragmentation event. So you fragment your fragment ions, your daughter ions, to, if you want, to your granddaughter ions. And you gain sensitivity by accumulating the fragments, and you gain specificity by the secondary MRM transition. So it's in principle something like MS to the 3, and therefore it is called MRM cube or MRM to the 3. And this really helps increasing sensitivity, as I'd like to show you with these data. These are samples beef containing pork or beef containing horse, and 2 specific marker peptides. And you do see at the given concentration we are at the limit of detection or even below the limit of detection in the MRM mode. The MRM mode is the left part of the slide, and on the right side of the slide, in parallel we measured the MRM cube trace at the exactly the same concentration, the identical sample measured in parallel, and you do see that signal to noise is really vastly increased, and we are really nicely able to detect 0.2% pork in beef, for instance. We are currently able, using this technique and by further optimizing the extraction procedure, to detect something around 0.05% of pork or horse in beef, and this also works out in general in processed samples. So by using MRM cube you really can gain sensitivity, and I'd like to show you later on with the allergen detection that you also can increase specificity. This approach also works in processed samples as long as you are able to extract your proteins. We first of all, as I mentioned, used buffers for partial proteome extraction, so in general high salt buffers to extract the myofibrillar and the sarcoplasmic fraction of your samples. This is shown here, buffer 1, and you do see that in raw samples you do see the respective myofibrillar and sarcoplasmic proteins, so it's not a complete extract, but the proteins of interest are extracted. But after thermal processing, frying, cooking, other heating steps, we were not able to extract anything. And by switching to high urea buffers, we were then able, here given on the right side, we are then able to really perform nearly complete extraction of the proteins given in this sample, so vastly increased extraction efficiency in the raw samples as well as in the processed sample. And this really increased the detection limit and also allowed us to detect our peptides of interest in processed samples. So we used different processed samples. There's just one example here. The upper lane is one corned beef sample which we bought in a German supermarket, which obviously contains horse meat, as shown by the specific markers identified for horse in MRM and MRM cube. The lower lane is another corned beef sample which we also bought and which is obviously devoid of horse. So this approach obviously also works in processed samples such as corned beef and in commercially available samples. So we were quite happy to see that, and we then moved on further recently to analyze really highly processed samples. And one example are the so-called processed animal proteins, or shortly PAP, which are used for feeding purposes. And if you just have a look at the gel here on the right side, you do see that for pork meat, we are able to extract using high urea buffer. So this is the buffer composition which we use for our processed samples, and meanwhile also for our raw samples to get highest extraction efficiency. In this pork meat samples, we were able to extract all the proteins, and if you look to the right, then you do see that for these PAP samples, you do not see any protein bands any longer because of this high thermal processing and this really prolonged thermal processing. We were not able to see any protein bands, but we did still see the peptides on peptide level. And the sample which we have analyzed contains 99% pork and traces of chicken or poultry in general, and we were able to detect two chicken markers here given at the lower part of the slide. One marker with 3 MRM transitions still, so minimum quality criteria fulfilled, and the other marker we were also able to detect with even 6 MRM transitions, so we did not even use MRM cube, and we were still able to detect from this processed animal proteins these markers in trace amount. So we were not completely sure how the amount was, but it was expected that we have at least 99% pork and something below 1% chicken or other bird species. The next step was then to switch to multi-method to extend the number of species we are able to detect, and we developed a method including 9 different mammalian and also bird species. We have identified species-specific marker for all species, and we are now about to publish these results. It's pretty recent work. We also were able to identify general markers for the detection of animal and group-specific markers, for instance, for the detection of all different deer species. We were able to differentiate fallow deer, red deer, and roe deer. We have specific markers for these species, but we have also group-specific markers which we can use to detect all deer species. And the idea behind these group-specific or general markers and the species-specific markers is that even if you have contamination or food fraud with exotic species or species which you do not expect in your sample, you will be able to see that something is wrong with your sample because the signal ratio between your specific markers and your general markers will change when you have addition of exotic species where you do not have specific marker for, but you can at least see that this sample obviously is not okay, and might then use other techniques to go more into detail and to analyze this sample more into detail. So this is the part concerning food authenticity, and the next part I'd like to show you some of our work on allergen detection, also using mass spectrometry. But before we have a look at the MS data and the approaches we have used for allergen detection, I'd like to give you again a brief introduction on food allergy. In general, food allergy is a misinterpretation of food proteins by the immune system. So the immune system is misled and develops a specific immune response against completely harmless food proteins, and develops specific immunoglobulin E or IgE antibodies, and this process is called sensitization. And once you are sensitized against a specific food, peanut for instance, and you then take up this food again, you will see binding of the food allergen or fragments of the food allergen on the surface of mast cells to the specific IgE antibodies, and this then leads to degranulation of the mast cell, release of highly potent mediators such as histamines, leukotrienes, prostaglandins, and others. And these mediators then lead to the allergic symptoms. If you have a mild food allergy, it's itching in the oral cavity or sneezing, and it can also be a number of different gastrointestinal symptoms, and it can be even life-threatening or fatal complications like the anaphylactic shock. And the prevalence of food allergy in Europe is something around 2 to 4% of the adults and 5 to 7% of children, which do suffer from IgE mediated food allergy. So this is approximately 17 million citizens in Europe that are affected by a real food allergy, and the numbers are similar in North America. In the US, we have something around 8 to 9 million food allergics, and the prevalence is similar, something between 2 to 4% of the adults and something around 5 to 7% of the children in the US also suffer from food allergy. In the US, there are, in addition, data on the fatal incidences when food allergics take up accidentally the allergenic food. And there are estimated that there are in between 150 to 200 deaths per year in the US just by accidental uptake of food allergens by the allergic consumer. So this is really a problem for food sensitivity, food safety, and this is really, in industrialized countries, really one of the major problems, at least from my point of view, concerning food safety. Having a look at the allergenic food, there are quite a number of different foods that have the potential to elicit allergic reactions. 170 to 200 different foods can lead to allergic reactions, but only a very limited number of different foods regularly cause problems. It's the so-called Big 8: wheat, fish, peanut, milk, tree nuts, soy, the different crustacea, so lobster, shrimps. The major point is that we are now about to develop suitable analytical techniques to really identify the allergens in the food and to control the food by use of analytical techniques and not by may-contain labeling. Basically, the idea is identical with the identification of peptide markers concerning food authenticity. The major food allergens is an additional criteria. So we do not want to detect species, hazelnut or peanut. We want to directly detect the eliciting agent, the allergenic protein, and therefore we focus only on these proteins. And in addition, we want to be able to distinguish specific food allergens from different species. If you have a look at the allergens, it is in general the case that you will see families of food allergens, the 11S legumins, for instance, as seed storage proteins, which you do see in hazelnuts, in pecan, and in walnut as well. And they do have really incredible high sequence identity, something around 95%. So what we want to do is to distinguish the 11S from pecan, from walnut, and from hazelnut. And this is one thing where ELISA, for instance, from time to time fails because of this incredible high sequence identity. And this was one of the further criteria we wanted to address. So the basically approach was similar or identical compared to the food authentication. But we started with a far lower number of peptides as we only focus on the allergenic proteins, but ended up with a number of suitable markers for 6 different nut species. So we, in the first study, focused on the different nut allergens: hazelnut, cashew, almond, peanut, pistachio, and walnut. And then as we really do need to be really sensitive, as the eliciting dose of the most sensitive allergic patients is incredibly low, with something around 0.5 milligram total hazelnut and something around 1 milligram total peanut. We really needed to be very sensitive, and therefore we further optimized in a systematic fashion the MRM cube approach and tried different parameters. And the major parameter which had influence on signal intensity and sensitivity was the fill time, which is not too surprising. So what we actually did, we prolonged the fill time in the Q3, in the third quadrupole, filling up with the fragment ions, which we can then use for further fragmentation and for detection using the MRM cube approach. And you do see here the different filling times for specific fragment ions and MRM cube transitions. And one of the nice things is that we did not see really overfilling effects, which can occur if you fill up too many ions. This is something we did not see in our study. So we prolonged the fill time from the standard setup 50 milliseconds to 250 milliseconds, which is the maximum fill time which is possible. The downside is that the cycle time also is increased. So if you have really a multiplex analysis, you will face the problem that you have already 250 milliseconds fill time for every MRM cube transition, so you will have pretty long cycle times. And we solved this, or surrounded this problem, by dividing our method in different periods, and we have only in the different periods we have only a limited number of peptide markers which we detect using MRM cube. And maybe this is the situation to directly address SciEx and to ask whether it might be possible to also develop scheduled MRM cube, which would be fantastic because you can then use this scheduled approach, which is already available on MRM for MRM cube, and can then really perform multiplex analysis of high numbers of different food allergens or species, for instance. We then checked the method performance using MRM cube and compared this with the normal MRM approach. We spiked the different nuts at different concentrations in 3 different matrices: bread, ice cream, and chocolate. And we were really surprised to see that we are pretty sensitive. This is one example where peanut peptide was spiked in bread, and we were even able to detect 0.3 microgram peanut, not the specific peptide, but calculated as peanut per gram bread, which is really sensitive and which is well below the sensitivity we do need to protect also the most sensitive allergic consumers. And if you compare it with the MRM trace, which we in parallel also measured in these samples, you do see that we are below the limit of detection and that we are somewhere in between 5 to 10 microgram peanut, in this case, per gram bread. We were able to detect the peptide also using MRM. So again, really massive increase of sensitivity, and we observed something around like a factor of 20 when using MRM cube with a prolonged fill time compared to MRM. On this slide, I'd like to show you that specificity can also be increased using MRM cube, and this is exemplified by one example where we have here an almond peptide in ice cream matrix, and the upper lane on the left and the right is again MRM cube. On the left you do see the blank sample as it should be, no signal for any of the MRM cube transitions. On the right side a spiked sample where you do see both MRM cube transitions, and the lower lane here is in parallel again measured the MRM trace, and you do see in the blank sample at the correct retention time signals from 4 MRM transitions in the correct signal intensity ratio. So this lower blank sample would have been a false positive when only using MRM, and this obviously can occur even when you identify really specific peptides in some matrices, and therefore MRM cube can really help and can really improve specificity. On the right side, I'd like to briefly show you that the MRM approach also needs to be used with some caution. This is a walnut peptide in different matrices, and again you can see here on this upper lane, these are 3 different MRM cube transitions, and the first MRM cube transitions in bread matrix, here you do see at shifted retention times unspecific signals, and this is obviously due to the fact that we have used pretty small fragment ions. So the first fragment is a so-called Y2 ion, a deep peptide if you want so, and this is further fragmented to the Y1 ion, which is a free amino acid if you want so. And the smaller the structure of the fragment ions is, the more reduced is the sensitivity. So my advice would be to use MRM transitions for MRM cube, which are not too small. If you look at the middle and the lower lane, you do see identical sample, identical peptide, and you do not see any unspecific signals. So this is just one brief advice that you should not use too small fragment ions in MRM cube. We use this approach also for commercial samples. This is just one example. This is a spiked chocolate where we do see 3 hazelnut-specific peptides with 2 MRM cube transitions each. So for MRM cube, we decided, or we identified that we do need 2 MRM cube transitions to be specific. And the lower lane is a commercial chocolate as one example that we were nicely able to detect all 3 hazelnut allergen-specific peptides with all MRM cube transitions. We also started a project on identification of Crustacea, and this is somehow a link between allergen detection and species authentication, because these marine foods are prone to food fraud as they are rather costly, and there are in addition major allergens. So again, you have both aspects, authenticity and food safety for these foods, for the Crustacea. And we again used the MRM cube approach with really high sensitivity. And again, MRM cube really improved the limit of detection compared to MRM. In general, the limit of detection was higher compared to the nut species, and this is due to the fact that the amount of allergens in Crustacea is far lower than the amount of allergens in nuts, as in nuts mainly the seed storage proteins are the major allergens, but the eliciting amounts which are necessary to elicit a reaction are as well increased in Crustacea compared to nuts. So we are again well below the necessary sensitivity using this method. On this last slide, I'd like to give you a very brief idea on the factors that might influence quantitation once it comes to absolute quantification. So the aim finally is to have something like risk-based management of food allergens using threshold levels, which need to be identified, and there are really studies around in Europe, the Europreval study, for instance, where the eliciting dose for individual allergic patients is identified in a clinical setting. So we now have really an idea what the eliciting doses and thereby the amounts in food are which we need to detect. And in principle, we are now underway that we might define such threshold level. Not very much surprising, the food matrix as well as processing and sample preparation can affect absolute quantification. We have started with a first study on our hazelnut and other nut allergen markers. And we observed that there are different aspects which need to be taken into account. One is, again, extraction of the proteins and digest of the allergens, which can be affected by the food matrix. And further on, the released marker peptides can also be influenced by the food matrix. And the latter point can nicely be controlled by use of stable isotopically labeled peptides, which are in principle available. They can be synthesized commercially and in principle can be used. But future studies, in my opinion, should also focus, and we will in future studies focus on the matrix effect on protein levels for the allergens. So we might need matrix-matched correction factors that take also into account extraction efficiency and release of the peptides from the allergenic proteins. And this is just one of the outlooks what need to be done if we want to end up with the really absolute quantification, not also detection of allergens, but also really absolute quantification of allergens in food. We need to take into account these aspects. And with this, I'd like to thank you for your kind attention, and please excuse that the technical problems we have observed here. I'd like to thank the sponsors from our research, SOIX, the DFG, the European Union, and the Federal Ministry of Food and Agriculture. And I'd like to specifically thank my group, and especially Robin Korte, who is now at the moment presently finalizing his PhD thesis, and he was the one who did most of the work I presented today. Thank you very much for your attention.
Thank you, Dr. Brockmeyer, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Brockmeyer will answer as many questions as time permits, and the first question is, why mass spec and not PCR or ELISA?
Basically, when it comes to food authentication and not food allergen detection, PCR, of course, also has its place and also, of course, has worked in the horse meat scandal, but there are specific problems with processed and really highly processed samples. So the processed animal proteins, which I've presented in my presentation, We obtained these samples from research group that worked on PCR detection and really told us we do not see anything using DNR-based techniques. Are you able to see anything in these samples? So when it comes to food authentication, in really highly processed samples or samples where acidic conditions play a role, I think that mass spectrometry is really a very nice alternative as we really can detect in these highly processed samples still our peptides. And we can quantify and relatively in an absolute fashion also quantify using these peptides, which is another advantage. When it comes to allergen detection, I think that mass spectrometry is the ideal solution as we directly detect the eliciting agent, the food allergen, by detecting really a specific structure. And this is the problem with PCR. You detect the DNA and not the eliciting agent. Again, by processing, there can be really problems that DNA and proteins are degraded not identically, and therefore I think that here the direct detection of the allergen is the best solution.
Is there any opportunity to speed up the sample prep time?
Yes. At the moment, we are about to do some work on speeding up the sample prep time. We have also improved already the extraction time. So the extraction time at the moment is at about 1 minute, which is pretty quick, I think. And we are about to optimize the digestion time. This was one of the parameters which I briefly mentioned at the end of my talk in the last slide, where we also looked at the optimal digestion times and how can we optimize and speed up the digestion time. You can also use online desalting prior to mass spectrometry, which would also really speed up sample preparation. And we will have a look at techniques such as microwave-assisted digestion, where you really can, under ideal conditions, reduce digestion time from several hours to several minutes, and this would really help to speed up.
How long does it take to do the peptide digestion?
At the moment, the peptide, we have presently in our study not yet really focused on speeding it up. So the general protocols which are around from the proteomics laboratories or the biochemical laboratories are often simply overnight, and to be honest, most of the time nobody really looked at how the efficiency is. We reduced this time to 4 to 6 hours, and using techniques such as already mentioned, such as microwave-assisted tryptic digest, and there are also other techniques around. It is in principle possible, and there are studies already published, that we can reduce the digestion time to something like 10 minutes, 15 minutes maybe. And then the sample prep would be pretty quick. So this is basically the idea, to speed up this specific part of sample preparation.
Which kind of allergen source do you use to spike the allergenic food into the matrix, for instance, flour or food?
We used, for the bread matrix, for instance, we used flour. So we baked the own bread and used the normal wheat flour, for instance, and spiked the hazelnut flour in the sample. We also used from time to time really milled or rather coarse flour, so particle size was increased, but we did not use, for instance, really peanut pieces or hazelnut pieces, because you of course then always have the problem of sample homogeneity, but this is a rather general problem and not a specific problem for mass spectrometry. If you have particulate contamination, such as hazelnut pieces or peanut pieces, which are not in the complete food you analyze, but only in one part of the food. This is a general problem where you really do need to take huge sample amounts and homogenate the samples to then analyze these samples. But for the study I have provided you, we used flour mainly.
Where or how do you feel this technology compares to whole genome sequencing?
Whole genome sequencing is relevant in authenticity testing. Actually, it does not, in my opinion, not help concerning allergen detection. You can perform species identification, but again, you have the problem that you detect DNA when you want to detect eliciting agent which is proteinaceous. But of course, there are some laboratories around that now start next generation sequencing as a detection tool for species authentication, and this is indeed promising when you have food which is not incredibly complex, and if you do not want to precisely quantify. So one of the problems, at least as far as I can see, with next generation sequencing is if you want to really detect minute amounts and quantify minute amounts of a given species in a complex food. This, as far as I can see, can be a problem. But on the other hand, next generation sequencing is of course as well pretty powerful technique when it comes to species authentication.
Typical MRM experiments are quantitative. Are MRM cubed experiments also quantitative?
Yes, they are. We were really positively surprised that MRM cube, as long as you do not overfill the linear ion trap, they are quantitative as well. So it was also shown on one of the slides where you did see the correlation curves for the different concentrations, and they were pretty similar for MRM and MRM cube. So as long as you do not start and if you have really incredible high amounts of your analyte, you will end up overfilling your trap. But this is at the end of the day also a problem if you have far too high concentrations with MRM. So as far as we yet have seen in our studies, you can use it nicely for quantification. It's quantitative, yes.
How can you correlate the allergen protein amount to the food amount in grams?
In our study, we did it by spiking, and this is exactly the problem which we have to solve, and which what I addressed on the last slide. So the absolute quantification. What we of course can do is, if we have an example for the hazelnuts, one thing we will do also in the future is to see whether the allergens, and these are mainly seed storage proteins, whether the amount of the seed storage proteins is stable within the hazelnut, so X% of the total protein in the hazelnut is allergen A or allergen B. And you can from that then delineate from the identified peptide from the allergen to the from peptide level to the protein level, and then calculate from the protein level to the food level. One of the other points is that if we have methods around to detect the food allergen, it is not really necessary to calculate hazelnut. This is actually has been developed because we weren't able to directly detect the food allergens, and what DNA and PCR-based method actually do is that you have a species identification, hazelnut, and then you calculate the amount of hazelnut because you're not able to detect the allergen directly. So my idea for the future would be that we have no threshold levels for hazelnut, but we have a specific threshold level for the given hazelnut allergens, similar to mycotoxin levels or pesticide levels. And then we do not need to have these calculations because we directly calculate the eliciting agent. And this is pretty easy to do from the peptide level to the protein level.
Looks like we have time for one more question, and it will be, the use of animal group specific markers sounds very interesting. Do you think that this can be put into routine testing?
Maybe you can
authenticity testing?
Yeah. I think that this is really possible. We have contact to a number of laboratories which are really interested in implementing the authenticity testing in routine. And I do not see any point why this shouldn't be possible. Actually, we started developing this method to translate them in routine analysis. So basically the idea is that we as an academic research group identify the peptide markers, transfer the method to machines that are around in routine laboratories, triple quadruple instruments, Q-trap instruments, so that these instruments can be used. And we are actually in contact with different laboratories, with different stakeholders that are also really interested in implementing these methods in routine. So yeah, I'm really sure that this is possible and that this is feasible, and I'm happy to see this actually, because this would mean that our research then is translated into routine food authenticity and food safety testing.
I would like to once again thank Dr. Brockmeyer for his presentation. Do you have any final comments?
Actually, I thank you all for your kind attention, and I really do hope that you had an inspiring talk and get some ideas on food allergen testing and food authenticity testing and what is nowadays possible using mass spectrometry. And I'm happy also to take your questions via email if you have any questions to discuss aspects such as how can this be translated into routine analysis. So yeah, thank you.
Thank you, and I'd also like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would also like to thank our sponsor, SciEx, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through November 18th, 2017. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.